Now we're going to go step by step through the process of DNA replication in detail. The first thing to understand is that eukaryotic chromosomes, so humans, are linear, which means that they're one straight line and they don't form loops. What happens, instead of starting at one end and going all the way to the other end, which would take a really long time, is replication happens along the entire length of the chromosome all at the same time. So along this chromosome piece, we can see that there are 3 regions here. This is the double-stranded DNA. There are 3 regions here called the origin of replication. The origin of replication is where we're going to focus in on the details, but there's tons of these happening along the DNA strand. So here you can see this is one replication bubble, here's another one, and here's another one. And this is one long strand of DNA. So at each origin of replication, the DNA helix is going to split apart, and we're going to actually add DNA going in both directions. So you can see that replication goes this way and this way. Eventually, these replication bubbles are going to meet. When all of the replication bubbles are done, you end up with 2 distinct strands of DNA. So it doesn't just start from one end and go all the way to the other end. It starts in little bubbles all along the process. So what we're going to do here is we're going to focus in on a replication bubble and see what's happening here at the molecular level. So 1/2 of a replication bubble is called a replication fork. So right here would be the origin of replication. This would be where the replication starts. And then there's the rest of the bubble is on this side. So we're just going to look at half of the replication bubble. And that creates this Y-shaped region called the replication fork. So that's right here, where the parent strands are being taken apart. The very first thing we have to do is we have to separate the parent strands. The very best way to learn DNA replication is to start by learning all of the enzymes. There are going to be a ton of enzymes involved in DNA replication, so you need to make flashcards or a table or something and learn all of the enzymes and then come back and study the process. So the first enzyme you need to know is DNA helicase. DNA helicase unzips the DNA strand. So if you look at this like a zipper, here's DNA helicase right there at the replication fork, opening up the 2 strands. Once this is open, it's going to tend to kind of collapse back on itself and stabilize. A single-stranded DNA is not a very stable structure. So the second enzyme that you need to know is the single-stranded binding proteins. You can abbreviate these as SSBP if you'd like to, single-stranded binding protein. So what these do is these form on the single-stranded DNA in order to keep them separate. So this is going to prevent the DNA from coming back together. So remember that DNA is a helix. So this is actually a twisted structure. Think about this like curly hair. Have you ever tried to run a brush just straight through curly hair? It doesn't work very well. The brush just gets caught in the tangles. What you have to do is you have to untangle it first if you're going to try to run a straight brush through it. So same thing with DNA. The DNA is a helix shape. You can't just take 2 strands that are twisted around each other and pull them apart. What's going to happen is you're going to get supercoiling and twisting. So the third enzyme is called topoisomerase, and this is going to be upstream, meaning that it is where the DNA replication is headed. So this is before the replication fork hits this. So it's upstream in the DNA. What it does is it cuts one piece of the DNA and it allows the DNA to untwist. So if you've ever had something that was really twisted, one way you could untwist it is just kind of hold it and let it untwist on its own. So that's what topoisomerase does, is it's going to let this region untwist on its own so that when helicase tries to open it up, it doesn't cause tangling of the DNA strand. So these three enzymes are really the setup before replication gets started. The very next thing we're going to have to do is start adding some new nucleotides. So now that we've got the DNA replication bubble, the origin of replication is open, we want to go ahead and start adding the complementary nucleotides. However, DNA polymerase, which is the one that adds nucleotides of DNA, has a limitation. DNA polymerase cannot start with nothing before it. DNA polymerase can only build off of an already existing molecule. So before we can add DNA, we actually have to add a short RNA primer. So here's my next enzyme, which is primase here. Primase is going to add a very short, so this one is literally 5 nucleotides long, piece of RNA primer. So in red here, we can see that we have started at the origin of replication. Whatever this molecule is, RNA primase is going to add the complementary RNA a short distance here to make a small RNA primer. This RNA primer is just a temporary thing. It's not going to last. It's not going to stay. We will eventually have to replace it. Now that there's a primer in place, then we can start to add DNA. And you can see the next enzyme right here is DNA polymerase 3 that's going to add the DNA from the existing primer. And we're moving in this direction. We're going to be moving from 5' to 3'. So here's my 5th enzyme, DNA polymerase 3. There are 2 different DNA polymerases, so you need to know which one is which. DNA polymerase 3 is going to add DNA nucleotides to the primer to the growing DNA strand. When it adds nucleotides, it actually adds them as a nucleoside triphosphate, and it breaks these phosphates off to produce the energy needed to build this molecule. So this is a dehydration reaction. When this is complementary base pairing, so you can see that this is the template strand, and here's the anti-parallel strand. The orientation of this triphosphate matters, so it has to add them. We can only build DNA in the 5' to 3' direction on the new strand. So here's the new strand. DNA replication starts up here at the 5', and replication is going to proceed this way into the 3'. So 5' to 3' direction, you need to memorize that. DNA replication always goes 5' to 3' on the new strand. So here we can see that the next nucleic acid that's being added is the T to match with the A. The next one here will be a G, and then it will continue to proceed this direction. Eventually, once we have added all of the DNA molecules, we need to go back and replace the RNA with DNA. So the 6th enzyme here is DNA polymerase 1. DNA polymerase 1 comes back at the end and removes the primers and puts in DNA now, building off of the DNA that has been built up to the primer. All right, so we now know that DNA can only elongate in the 5' to 3' direction. But we also know that DNA is anti-parallel. So when I have 2 strands of DNA, if this prime side runs 5 to 3, this side runs 5 to 3, the opposite direction. So when I build the new strand, I'm going to have to build them in different directions. So let's label these down here. This new strand is being built in the 5' to 3' direction. This one is being built in the 5' to 3' direction. Remember, for each arrow, 5 to 3, 5 to 3, 5 to 3. Whenever the 5' to 3' direction is going towards the replication fork, so right here and right here, the replication is easy. It can start at the origin of replication at 5', and it can move towards the replication fork, and as this keeps opening, it can just keep moving forward. That is called the leading strand. The leading strand is built in one piece. So they add the primer. Here you can see the RNA primer, and then DNA polymerase III will come in and add all of this DNA. And then eventually DNA polymerase I will come in and remove the primer and put in the DNA. So that works really great for half of the replication bubble. The other half of the replication bubble is called the lagging strand. This is going to physically take longer and be slower. So replication is proceeding outwards towards the replication forks. However, for this lagging strand, 5' to 3' is going the opposite way. 5' to 3' this way. 5' to 3' is that way. So what has to happen is the replication fork opens and it makes a short piece. And then when the replication fork opens a little more, we can move further into the replication fork and make another short piece. And then again, the replication fork opens a little bit more, and you can move further into the replication fork and make a short piece. So the lagging strand gets made in pieces called Okazaki fragments. Eventually, these fragments have to be put back together. So here's enzyme number 7, DNA ligase. Remember, ligate or a ligand means to bind. So DNA ligase is going to come in at the end and connect all of these little fragments back together into a long strand of DNA. So now we're going to take a closer look at what's happening on the lagging strand. So here's my lagging strand. Here's my origin of replication. That means that replication is proceeding in this direction. Here's my replication fork over here. But I have to go 5' to 3'. So helicase already opened this. Single-stranded binding proteins, they're way over here holding it apart. Topoisomerase is over there holding it so that it's untwisting. RNA primase here has added a primer. This is the primer for the next leading strand. So here's the origin of replication. It's actually right there. So here's the first primer. The replication fork is opened enough that primase can come in and form a small primer here. Next, we get DNA polymerase III is going to go ahead and start here from this primer and add DNA in the 5' to 3' direction, which is going to head towards the origin of replication. Once that's done, the DNA polymerase can detach, and we have our first Okazaki fragment. So this is the fragment number 1, and so you see they've labeled it number 1 because it's close to the origin of replication, which is right here. Now we can continue to move over here to the left. Remember, the replication fork is over here. At this point, it will have opened even further. So before it might have been open this big, now it's going to be open wider. So we're going to be able to move further into the replication fork here. So we can move further into the fork. We're going to make another primer, so another RNase primer. And then DNA polymerase III will come in, add DNA to make Okazaki fragment number 2. Over here, you can see this was primer number 1 and fragment number 1. DNA polymerase I will come in here and start to replace the RNA with DNA. So now you can start to see it's once Okazaki fragment number 2 is done, DNA polymerase I can actually start at Okazaki fragment 2 and go in the 5' to 3' direction and add DNA and remove the RNA. We cannot have RNA left in our DNA at the end. We have to remove it all. So that's going to leave us here with 2 fragments. Notice that there's a small cut between those 2 fragments where they are not connected. So the very last thing we need to do is have DNA ligase, or just ligase, come in and form a bond there and go ahead and close that up. So now Okazaki fragment 1 and Okazaki fragment 2 have been bonded together. So the overall direction of replication is going towards the replication fork, but because each piece has to be built 5' to 3', we have to do it in small chunks. So here's the whole entire overall replication. We can see the origin of replication is right here at this end. So this is the origin of replication. And then over here we can see the replication fork. This bubble is fairly large. Replication has been going for a while. Let's start at the beginning. Number 1, helicase is going to open up the replication fork. Helicase is going to proceed towards the left here to continue to open this replication fork. Number 2, we have the single-stranded binding proteins, or SSBP, that are going to follow helicase and just continue to hold that replication fork open. Enzyme number 3 would be topoisomerase, which is very far upstream, helping to untangle the DNA as this replication proceeds. Let's look at the leading strand first. At the leading strand, we're going to add a primer with primase. So that primer would have been over here. And then DNA polymerase 3 is going to continue to add DNA in the 5' to 3' direction. And so you can see that this is one long strand of DNA, and it's going to just continue to proceed towards the replication fork. So the side where 5' to 3' moves towards the replication fork is called the leading strand. Eventually, DNA polymerase 1 came in and cleaned out this primer, and then ligase binded this, and now we have one nice long strand on the leading strand. On the lagging strand, we have to do these steps kind of in concert. So you can see that it's working on Okazaki fragment over here with a primer on primase. DNA polymerase 3, this is enzyme 5, is over here adding DNA to Okazaki fragment number 4. Okazaki fragment number 3 was finished, so the DNA polymerase number 1, so that's enzyme 6 here, is changing out the RNA and turning it into DNA. And it has to wait for this fragment to be done because it has to be able to use DNA as a starting point. DNA polymerase cannot start in thin air. That's the whole reason we have the primers. And then eventually the last thing that's going to happen is DNA ligase is going to bind these two together so that we have a nice strand. So you can see how this lagging strand is probably going to take longer than the leading strand because there's a lot more steps involved, and they have to make a bunch of these small Okazaki fragments.